Molecular basis of smooth muscle contraction. Calcium is involved in the initiation of contraction of smooth muscle. However, visceral smooth muscle generally has a poorly developed sarcoplasmic reticulum, and the increase in the intracellular calcium concentration that initiates contraction is primarily due to the calcium influx from the ECF via voltage gated and ligand gated calcium channels. In addition, myosin in the smooth muscle must be phosphorylated for activation of the myosin ATPase. In smooth muscle, calcium binds to colmodulin and the resulting complex activates colmodulin-dependent myosin light chain kinase. This enzyme catalyzes the phosphorylation of the myosin light chain on serine at position 19. The phosphorylation allows the myosin ATPase to be activated and actin slides on myosin, producing contraction. This is the contrast to skeletal and cardiac muscle, where contraction is triggered by binding of calcium to troponin C. Myosin is dephosphorylated by myosin light chain phosphatase in the cell. This enzyme is inhibited when it is dephosphorylated and activated by dephosphorylation. It is dephosphorylated by Rho-associated kinase that is activated by ligands which produce inhibition of smooth muscle activity. However, dephosphorylation of myosin light chain kinase does not necessarily lead to relaxation of the smooth muscle. Various other mechanisms such as latch bridge mechanism, by which myosin cross bridges, remain attached to the actin for some time after cytoplasmic concentration falls. This produces sustained contraction with little expenditure of energy, which is especially important in vascular smooth muscle. Relaxation of the muscle presumably occurs when there is a final dissociation of calcium colmodulin complex or when the other mechanism comes into play. The events leading to contraction and relaxation of smooth muscle are summarized below. Initially, binding of acetylcholine occurs to the muscarinic receptor, which causes increase in influx of calcium into the cell. Calcium binds to colmodulin. There will be activation of colmodulin-dependent myosin light chain kinase, which leads to phosphorylation of myosin. Increased myosin ATPase activity and binding of the myosin to actin occurs that leads to contraction. Then dephosphorylation of myosin light chain by myosin light chain phosphatase occurs, which causes relaxation or sustained contraction due to the latch bridge and other mechanisms.